Hello, welcome to another session. In this session, I am discussing about the process of gastrulation in human. So, what is the process of gastrulation? So, by the end of first week of fertilization, blastocyst is formed, and by the second week of fertilization, implantation is taking place. Implantation is over, and by the beginning of third week of fertilization, the process of gastrulation starts. The process of gastrulation begins by the end of third week of fertilization. So what is the meaning of gastrulation? Discussed earlier that the blastocyst, which also known as the blastula, this is known as the blastula. The blastula, it has two mass of cell, the outer mass of cell, which is known as the trophoblast. So this is the outer mass of cell. This is known as the trophoblast. And inner mass of cell, which is known as the embryoblast. So these cells are known as the embryoblast. So these are known as the embryoblast. Gradually, such trophoblast, which divides and which forms two types of cell. Some of the cell which present outside, or some of the cells generate by the division of the trophoblast cell outer side, and such cells are multinucleated, and these cells are known as the syncytial trophoblast cell. So the trophoblast cells which divide outside and they form the multinucleated cell or the syncytial cell, those are known as the syncytial trophoblast cells. And the inner mass of cells, now the trophoblast cells, now they are known as the cyto. These are the single nucleated cell. These are known as the cytotrophoblast cells. And these cytotrophoblast cells retain the boundaries, but the syncytotrophoblast cell, these are the multinucleated cell, and they divide and divide, and each grow and grow subsequently, and eventually it surrounds the whole blastula or the blastocyst. So this is not the discussion of this video. After this, the process of discussion of the gastrulation, I will discuss about the formation of extra embryonic membrane. At that time, I will discuss what is the fate of the syncytial trophoblast cell. Now come to the implantation process. So by the end of implantation process, such blastocysts completely enter into the endometrial wall of the uterus and implanted. So now blastocyst, it present by the end of the second week of the fertilization. Now the blastocyst which present inside the endometrial wall of uterus. Suppose that this is the implanted blastocyst which present inside the endometrial wall of the uterus. At that time, such embryoblast cell, these embryoblast cell which divides, or which these cells, the mass of cells, earlier they are present in a clump, but during the second week of the implantation, such clump of inner embryoblast cell or inner mass of cell, they arranged in two layers. Suppose that these are the two layers. They arranged in two layers. The outer layers of cell, these cells are columnar shaped cell. These are columnar shaped cell. There is no outer. The dorsal columnar shaped cells, these are known as the epiblast cell. And the inner side, the inner layer of cells, these are cuboidal shape of cells. These are cuboidal shape of cells, and these cells are known as hypoblast. So this is the ventral side and upper is the dorsal side. So the upper or the dorsal side cells, columnar shaped cells, these are known as epiblast, and the inner cuboidal line of cells, which is known as the hypoblast. And gradually these cells divides or they extend and they form a space. So the space appear on the in between the epiblast and the trophoblast. So this is the outer is the trophoblast or cytotrophoblast. I told earlier this is the trophoblast or the cytotrophoblast. These are actually cytotrophoblast. So the space appeared. So the space appeared in between. Suppose that this blastula somehow it grows larger in size by the absorbing the secretion of the endometrial wall of the uterus. So this is the growing blastocyst or the blastula. Now the inner mass of cells differentiated into two types of cells. The dorsal is the epiblast and the lower is the hypoblast. Now the epiblast cell, that is the appearance of the cavity, that is the appearance of cavity in between cytotrophoblast and the inner and the epiblast. This is the epiblast. So by the formation of epiblast and hypoblast, the space is appeared in between the cytotrophoblast and the epiblast. This space is known as the amniotic cavity. This space or the cavity is known as the amniotic cavity. This is known as the amniotic. This is known as the amniotic cavity. Now the hypoblast cell. This is the cuboidal set of cells. These are present as such. So this is the cuboidal shape of cells. These are the cuboidal at the ventral side. These are known as the hypoblast. This is known as the hypoblast. Now, by the division of cytotrophoblast, another layer of cells appeared below the cytotrophoblast. So now the amniotic cavity, it is lined dorsal side by the another set of cells, and these cells are produced by the division of cytotrophoblast, and these layers is known as, or these cells are known as the amniogenic cells or the layer. This is known as the amniogenic cells because these cells for the amnion. That's why it is known as the amniogenic cells. It generates the amnion. That's why it is known as the amniogenic cells. So now the amniotic cavity, which is lined dorsal side by the amniogenic cells and the ventral side by the epiblast cell, and the hypoblast cell, which present ventral to the epiblast cell. This is the bilaminar. This is known as the bilaminar embryonic disc. This is the bilaminar embryonic disc. This is the bilaminar embryonic disc. How such bilaminar embryonic disc which convert into trilaminar embryonic disc. This process is known as the gastrulation. So what is the meaning of gastrulation? So gastrulation, this is the process in which bilaminar embryonic disc, means epiblast and the hypoblast layer, which convert into trilaminar embryonic disc, constitute of ectoderm, endoderm and mesoderm. So this process is known as the gastrulation, and such process taking place by the movement divisions of the cells of epiblast as well as the hypoblast. So let's see how such process actually taking place. Let me erase this side. So we have the bilaminar embryonic disc. Now we have the bilaminar embryonic disc. So three germs layer, ectoderm. So three germ layers, ectoderm, mesoderm and endoderm are formed from the bilaminar embryonic disc, and these three layers present one by one, and this type of disc is known as the trilaminar. This is known as the trilaminar embryonic disc. So bilaminar embryonic disc which present in between two space, lower. After the formation of this bilaminar, the lower blastocele, after the division of and after the lining of the cells of the hypoblast, after the lining of the endoderm, this is the Yolk sac. This is known as the, suppose that by the division of cells of hypoblast which form the endoderm, I will tell there are several names and which surround the space. Now the blastocele, it is known as the Yolk sac. This is known as the primitive Yolk sac. This is known as the primitive or primary Yolk sac. So the bilaminar disc now present in between two cavities. The dorsal cavity is the amniotic cavity and the ventral cavity, which is the primitive Yolk sac or the primary Yolk sac. After the formation of bilaminar embryonic disc, there is the thickening of surface of the epiblast cell taking place, and which forms a ridge like structure, and which ends or which goes up to the middle of the disc, epiblast disc, and this is known as the primitive streak. This is known as primitive streak. This is known as primitive streak. So if you take the section in this manner, and if you observe on top, then what happened? So before going through this, now I am making a very simple picture. Suppose that this is the embryoblast. Suppose that this is the implanted embryoblast, and there is the, suppose that this is the bilaminar disc. This is the bilaminar disc. This is the epiblast layer, and lower to the epiblast layer there is the hypoblast layer. This is the hypoblast layer. Above the epiblast, the space of the cavity is the amniotic cavity, and below the hypoblast, this is the yolk sac. So, if you observe on top, then what happened? What you see? This is the disc-like structure. This is the disc-like structure. Now you are observing on top by the taking the section like this. So this is the section and you observing on top. What happened? One side, after the formation of bilaminar germinal disc, this is the germinal disc or the embryonic disc. After the formation of this embryonic disc, there is the formation of primitive stick. So the thickening of surface of epiblast cells occurs, and such thickening means the formation of ridge-like structure found on the surface of epiblast disc and which extends up to the middle of the disc. This is the epiblast. This is the epiblast disc, and which extends up to the middle and which forms a node-like structure, the thickening node. This is known as the primitive node. This is known as primitive node, and this ridge-like structure which is known as primitive stick. This is known as the primitive stick. So after the formation of primitive stick, the region, the pole from where the primitive sticks start to generate or the appear, this region is known as the caudal region. This is the caudal or the tail region, and opposite to the caudal region or the axis, which is known as the cephalic region. Cephalic region or the cranial region. So the formation of primitive stick which shows the axis of the body, and there is the another plate on the epiblast disc, and this plate is known as, which also present in hypoblast also, this is known as the precaudal. This is the precaudal plate. This precaudal plate which indicates the cephalic region or the cranial region. At the precaudal plate region, there is the formation of buccopharyngeal region, and at the caudal region, this is the anal or the cloacal region. So after the formation of primitive stick and the primitive node, this is the accumulation of the cells on the surface of the epiblast, and which forms a ridge-like structure, a stick-like structure. And after the formation of, suppose that you take, or if you observe one side, so what you observe, suppose that this is the epiblast. This is the epiblast layer. This is the epiblast disc or the disc of the epiblast, and this is the primitive streak, and this is the node. So this is the primitive streak and this is the primitive node. Below the epiblast disc, there is the hypoblast disc. Suppose that this is the hypoblast disc. This is the hypoblast disc, and these are the hypoblast cells. So what happened here? After the formation of primitive streak, there is the dying of the cells of epiblast. Please try to understand. After the thickening, after the formation of primitive stick, now there is the beginning of the dying of the cells present in primitive streak as well as primitive node. So what happened now? So by the dying of the cells start to dying and which forms a groove-like structure. A groove or the canal-like structure. This is the groove-like structure. And suppose that this is the groove-like structure. This is the groove-like structure. Now it is known as the primitive groove. This is known as primitive groove. And there is the mass dying of the cells in the primitive node and which forms the primitive pit. This is known as primitive pit. In case of chick or in case of birds, the primitive pit is known as Hassen's node. After the discoverer named, it is named as the Hassen's node. But in case of human, in other case, it is known as the primitive pit. After the formation of primitive pit, what happens next? The cells which are present at the edge, suppose that these are the cells present at the edge, they start to secrete fibroblast growth factor 8. They start to secrete fibroblast growth factor. Fibroblast growth factor 8. After the secretion of fibroblast growth factor 8, there is the division and the formation of epiblast cells taking place vigorously. So more and more cells are forms at the edge of this primitive groove, and these cells now suppose that this is the groove, and this is the edge. The side of the edge, the cells divides and which produce more and more cells. And these cells now fall into the groove, and these cells fall into the pit. Suppose that like a waterfall, the water which coming and which entered into a pit, which entered into a groove. So like the water fall in a waterfall, like this the cells now fall in a primitive groove or the primitive pit, and this process is known as the invagination. So the cells which grow more and more and which fall into the pit, which fall into the groove, Please try to understand. After the formation of primitive groove and the primitive pit, the cells present at the edge of the primitive groove, they start to secrete the fibroblast growth factor 8. By the influence of the secretion of fibroblast growth factor 8, the cells are present at the edge of the primitive groove. They divide and redivide, and which produce more and more cells. Now these cells, they lose the stable to stick there, and these cells now fall into the primitive groove, like the water fall in a waterfall. So like this, the cells which now fall or which enter into the primitive groove or the primitive pit, this process is known as invagination. Suppose that here the red color cell which come to the ventral side. Now the cells coming to the ventral side. So ventral side, at the ventral side there is the hypoblast cell. This is the hypoblast cells or the disc. They replace the hypoblast cells. And which forms the endoderm. Now the hypoblast cells, these are replaced by the coming cells and which forms the endoderm. This is known as endoderm. This is known as endoderm. And some of the cells present in between, in between two discs, and these cells which forms mesoderm. The cells present in between two discs which forms the mesoderm. So this is the process by which bilaminar disc which convert into trilaminar disc. So this is the endoderm, and the middle which forms the mesoderm, and the upper cells or the upper disc which is known as the ectoderm. And these three plates which form the trilaminar embryonic disc. So this is the complete of the process of gastrulation, which taking place or which completed by the end of 3rd week of fertilization. So this is the process by which this bilaminar embryonic disc which convert into trilaminar embryonic disc, and this process is known as the gastrulation. And this is the precaudal. This is the precaudal plate here. This is the precaudal plate here. These are known as 3 germ layers. Our different organs and different systems, different system and different tissues, all these tissues, some of the tissues organs developed from the ectoderm, some of the tissues organs developed from the mesoderm, some of the tissues and organs developed from endoderm. So these 3 layers germinate our whole body. That's why these are known as the 3 germ layers. That's why we are the triploblastic animal, because our body has 3 germ layers. At the time of development we have 3 germ layers. That's why we are the triploblastic animal. And all the organ system originated from the trilaminar embryonic disc, and this process of organogenesis not included under the higher secondary level. To reduce the length of the video, I have another video regarding the formation of extra embryonic membrane. So if you like, then click another video regarding the formation of extra embryonic membrane. Thank you for watching.